Hello everyone, this is Fiza from group 316. Now we will study about oscillation in glycolysis. The non-linearity in the kinetics of biochemicals enzymatic processes manifest themselves in a number of cases in the periodicity observed. The origin of oscillation in biochemical systems in on the whole understandable. They are determined by autocatalytics and it in the sense are modeled by periodic reactions in non-living things. Auto-oscillations in glycolysis are spontaneous, self-sustaining oscillations in the glycolytic pathway. Here are detailed explanation. Glycolytic pathway. We have these whole enzymes glucose 6 phosphate, fructose 6 phosphate, fructose 1,6 bisphosphate, glyceraldehyde 3 phosphate, dihydroxyacetone phosphate, 1,3 bisphosphoglycerate, 3 phosphoglycerate, phosphoenol pyruvate. This is the whole procedure of the glycolysis pathway. Periodic auto-oscillatory phenomenon are characterized of the process of glycolysis, the anaerobic conversion of 6-membrane sugar to tricarboxy acid leading to the synthesis of ATP. Auto-oscillation mechanism, feedback loops. Glycolytic intermediate inhibits or activate enzymes. Allosteric regulations. Enzymes exhibit allosteric properties. Substrate inhibition. High substrate concentration inhibits enzyme activities. Product activation. Glycolytic product activates enzyme. Key enzymes like fructokinase 1. Allosteric enzyme inhibits by ATP and citrate. Pyruvate kinase inhibits by ATP and alanine. Glyceraldehyde 3-phosphate dehydrogenase inhibits by NADH. Oscillation characteristics. Periodic changes in glycolytic flux. Oscillations in ATP, NADH and NAD plus levels. Synchronized oscillations in enzyme activase. Robustness to parameters variations. Mathematical modeling. Ordinary differential equations. Nonlinear dynamic and bifurcation analysis. Computational simulations. Experimental evidence like we have in vitro study using yeast, muscle cells and cancer cells. Fluorescence microscopy and spectroscopy. Metabolic flux analysis. Consequences. Enhanced glycolytic efficiency, improved cell adaptation to changing environments, increased resistance to metabolic stress, potential implications for cancer metabolism and therapy. Regulations. Feedback inhibition by ATP, NADH and pyruvate. Feed forward activation by glucose and fructose 2,6 bisphosphate. Allosteric regulation by calcium ions. Diseases like cancer. Altered glycolytic oscillations contribute to cancer metabolism. Diabetes. Impaired glycolytic oscillations affects the glucose homeostasis. neurodegenerative diseases, altered glycolytic oscillations linked to neurodysfunction. Future directions: investigating auto oscillation in disease state, elucidating molecular mechanism underlying glycolytic oscillations, developing mathematical models to predict and control glycolytic behavior. Thank you.